really trying not to break it, or maybe I'm really trying to. Hey, how's it going? Thank you for being here. I heard that was like a big thing today. He was like, I need to find a cooler on this day.
Yeah, and I'm actually, I work on Hackathon.
Oh shit! I gotta show you this. It's super easy to use and super fun. We never had any trouble with it. Yeah, right.
So, happy to field questions. Otherwise,
thank you again for being here.
Yeah.
You should
No, I'm not hanging out with school. Just here for tonight. I think the Googlers is going to be here tonight.
Alright, so my computer is on in charge. You know, I do have one question. What is the Wi-Fi? No, for you.
For me? I have no idea.
Oh, okay.
Yeah,
Alright, and then just, Sean, if you want to just sort of put me up, or whatever. Yeah, let's try to switch to VGA. They even have the PC adapter on their own. That is amazing. I have never, I speak all the time. Literally a couple of times a month. I've never seen it. Still, it is not. Hey! How you been, man? Good to see you! Small world. Yeah, no, I am super happy to be here. What is airplane speed? Alright, yeah, I should do this one.
Yeah. Upload is going to take down. That's good.
That talk's going to happen at 8:40, but it's just so people have something to look at.
My colleagues here.
Just to make sure it works for everybody else. So I think that that is actually so that is going to happen between stuff and the objective. Oh yeah, so people keep asking me so basically, if we can get a SVAI government location is, then we can get another night.
Started. Can you please find a seat somewhere? There's a couple more up here in the front. And what really is sort of more touching to me than anything is that this room is full. By the way, you're looking at these slides, but I am going to give this sort of crash course into bioinformatics around 8:40. So anyway, yeah, so I think we all know why we're here. We are here because we are software developers, biologists, bioinformaticians, and we are going to try to make a difference in the world, and that's amazing. So thank you all for being here. That's awesome. So I was approached a couple of months ago by SVAI, by Pete at SVAI, who said, "Oh, you run genomics hackathons for the NIH. Will you come here and do this?" And I said, there is nothing I would like to do more. So I'm very excited to be here. And so Pete is up first to speak. So Pete has nothing he wants to say. If you have a good time at the hackathon, please, if you would do me a personal favor, if you have a good time at the hackathon, if things are really working out for you, go say thank you to Pete, because he put an immense amount of work. I spent an immense amount of time talking to Pete on the phone about this, and I know he spent way more time talking to the others up here, so that's awesome.
Pete,
so,
do you drink? This is for you.
Thank you, wherever you are, over there. Yeah. Hopefully, all of you, I mean, one of the awesome things about these kinds of genomics hackathons is not only the social good we get done, but getting to learn stuff and also meet other people. So try over the course of that hackathon to meet as many people as you can. You really meet a lot of cool, like-minded people, and that's really amazing. I'll talk about some other sort of more extended networking opportunities later. Another sponsor we have is Excel.ai. So I don't know who is coming up to speak for Excel.ai, if anyone, but, oh, Laura. Okay. This is Laura. I also spent a lot of time on the phone with Laura.
Hi, everyone. Welcome. We want to thank you all for coming out today. I actually do want Pete to join me. Maybe you can encourage him if you want to start saying Pete. Yeah. So Pete's actually the reason that I'm here. He said that he really wanted to do a hackathon together. I had been to some of his previous events for SVAI, and so I was really grateful and fortunate that he invited me to participate and organize this event alongside him. So why don't you say hi, Pete, and just talk about what SVAI is.
Okay. So I like organizing these things behind the scenes. Definitely less of a presence on stage. So SVAI is an artificial intelligence oriented group that focuses on the intersection of artificial intelligence and life sciences. We started the group about a year ago, and transitioned fully into focusing on artificial intelligence problems for life sciences in about November. So one of the things that we've always wanted to do with this group is bring people together to really produce something together, to really work on something that's more than just an event series that talks about how artificial intelligence is being used in life sciences. So this is our very first hackathon. This is our very first collaborative event where we're aiming to produce something, and it's a really special event. Ona will talk more about the specific
condition.
Yeah, the specific data set that we're looking at tonight. But I came from a healthcare background, and so this is a really special event for me. I'm really interested in seeing how we can bring more folks from the AI discipline into biology, genomics, and life sciences. I feel like there's a lot of people from in the AI space right now that have a genuine interest in this, in genomics and life sciences, but don't necessarily have a pathway to access it, to learn how to work with genomic data, to learn how to work with some of these complicated healthcare data sets. So we'll continue down this path. We have a lot of fun things planned. There's a lot more things that I'd like to say about it, and I'll say them over the Slack channel, over Twitter during the weekend.
Round of applause.
I'd just like to note that Pete said not 3 minutes ago that he had nothing to say.
So thank you, Pete, and thank you again for joining us. So at SVAI specifically is a career development program to train artificial intelligence engineers. And so our reason for being involved in this program, of course, is these are the types of problems that we would like our engineers, our fellows that go through our program to also solve. We want them to get involved with real-world problems in healthcare, in life sciences, and every other industry as well. So if you want more information about that, you can just check out our website. But thank you very much for being here. We are happy to organize this with you and happy to answer any questions that you might have going forward.
You're welcome. I'm looking at you. I mean, you don't have to. It's if you want to. All right, if you guys can let him through.
So this is a little last minute. I wasn't supposed to be here, so that's why I have no organized thoughts. But welcome to Google Launchpad. So I work on Google Cloud Platform, and specifically ML Engine and TensorFlow. So any of you who are playing around with those things, come see me. If you have questions, I have nothing else to say. I have a past life in life sciences, so I'm excited to see the intersection. But yeah, thanks for being here. Thanks for checking out this space and doing cool events. I got nothing else.
If you see this guy, please walk up to him and say thank you. He was able to step in the last minute and be the requisite Googler in the building for tonight. So he gave up his Friday night to be with all of you. So there's that. So thank you. And apparently there's a couple other sponsors who wanted to say a few words. If any of you want to come up, take 30 seconds, say something. Hi. I'm supposed to limit you to 30 seconds.
Okay, great.
So,
That's fine, because this is also very last minute for me, no organized thought. So I'm from Gen Life, and we're actually in InsurTech, which is kind of a tangent to healthcare. But for us, if you think about the healthcare system, right, insurance has a role to play because who pays for healthcare, who pays for the access, and with all the innovation going on, who gets access. And so we're working right now as an R&D partner for major insurance companies across the world, helping them leverage new and emerging technology like AI, machine learning, blockchain, Not sure about that one, but we're trying to figure out, so how do insurance transform from this paradigm of hedging risk, pooling risk, hedging risk, and seeing it as a commodity, and how does insurance begin to view itself as, and being incentivized basically to help us live healthier and longer, because technically they have an economic incentive to do so. And so we're helping them to think about that in the future. So if you were to build an insurance company ground up from today, how would you leverage AI? How would you leverage machine learning to do that, to be more efficient and more helpful? And so we're always looking to make friends in the machine learning AI space. So if you have any questions, if you're interested to look at the healthcare problem from another perspective, feel free to talk to me, or better, talk to my CTO who can't be here today. So I'm happy to connect anyone. Thank you.
Thank you. Hi.
Hi everyone, my name is June. I'm from IndieBio. We're a startup accelerator devoted to biotechnology. We have a space in downtown San Francisco where we give $250K in funding and lab space and co-working space for 4 months, and we have a whole program that ends in a big demo day where companies pitch in front of a lot of investors. So if you have a AI-related or genomics-related or biotech-related company, please chat with me, and we would love to have you join our next cohort.
Thank you. Anybody else before we turn the floor over to Ono? Anybody?
Go, Ono!
Yeah, that's awesome. So basically, I'm going to run through the schedule for the next couple of hours. So essentially, we are going to have a series of talks. So first is Ono, but then I'm really excited also to have Marco Giovannini here from the Children's Tumor Foundation. That should be an amazing talk. They really represent a lot of individuals with a lot of phenotypes. And then we will have a couple other folks talk. Anita from iris.ai, and then I will talk a little bit about literature and genomics as well. And then we'll have, then we'll have Burton Earnshaw, and then I'll give sort of a crash course we're getting there, bioinformatics. And then we will get in our teams and start working. Remember that at 4 p.m. on Sunday, we are done. So this is a very intense event. We will go, go, go. But now, without further ado, here's Ono.
Hello, everybody. Welcome, first of all. I am really, really impressed by what has happened in the last couple of months. Like, I could not have imagined so many people hacking on my own genome data. Now, it feels a little weird because it's kind of not something you do every day, I guess. On the other hand, it also feels very normal because it's, sounds like it should happen. Like it's all you can do at some point, right? And someone was saying, like, you're sitting on your data. Why? Like, I guess not anymore. Yeah, I think I gave like about over 100 people access now. So if you don't have access yet, we'll set it up tonight or tomorrow morning. So Marco Giovannini, who's coming later, will talk about NF2 in detail. He actually flew in with his own plane, and he had to delay his takeoff because of the weather in Santa Monica. So, but he will be here in about 20 minutes. So really happy that he could join. He's one of the top researchers in NF2. So he will be here the whole weekend and help you guys out on that part of the deal. So yeah, like again, I'm very impressed by the community that showed up. I think there are people mostly like data science informaticians here, but there are also people from the field of biology. There are I think I'm not the only patient. I don't know, are there any other patients in the audience right now? I don't think so, but there will come at least one more tomorrow. So that's going to be exciting as well. He's also going to hack. Yeah, this might also not be your typical hackathon. I think a lot of hackathons are centered around more technology problems, but this is actually like, it's a real medical problem that causes biology issues and issues with your body. So it's going to be interesting to make that conversion from like a biological problem to a technical problem, so you guys can work on it. So I'm really excited to see what you guys come up with, what kind of novel approaches you may find during the weekend. So what you see on the screen is actually part of my data. Probably not for you guys, not very exciting because you see this all the time. But I think more and more people will be familiar with this picture as we move along. I want to share some of my journey with you and how I got here. I think it's going to be. I try to keep it relevant to get some context on the situation. So it started like here. This is a photo from a city called Wijk bij Duurstede. This is in the Netherlands. Maybe you hear it from my accent, but I am not from here. Yeah, so it's, you know, pretty small city, quiet, not much going on. And I want to share like a little story with you, like that kind of, I think, tells a lot about how my brain works. So I was once in the kitchen and I was cutting my nails with a nail clipper. And then my mother, she told me that I couldn't do that in the kitchen. I had to go outside on the balcony because the nails would jump all over the place, and she didn't like that. She didn't like to clean it up. And so instead of going to the balcony, I changed the nail clipper in a way so that the nails wouldn't jump away, because I was like, I don't want to go to the balcony. So when I'm confronted with the problem, I kind of try to fix it. Yeah, later I tried to patent it and we patented it. And I think I was like 12 years old or something. So that it's you know, that's kind of what I do when I encounter a problem. And the other thing, I got really interested in the internet in my teenage time. So I started a little internet company in '98 and had bought my own server, put it in a rack, hosted some clients, and yeah, if you were there in the '90s, you might remember how hard it was to share an image or a video with a certain file size, and it was really annoying with your dial-up connection. Actually, today I feel the same with genomic data. I don't know if you guys experience that, like transferring 250 gigabytes for a BAM file is kind of a headache. So I had to really think about that time with our dial-up modems. Nothing really changes, I guess. Then I decided not to continue in the internet because I thought there was no career in tech. Everybody will be able to do it at some point. So I went to Delft, which is this nice city. It's a little bigger than Wijk bij Duurstede. I think about 10 times bigger, 20 times bigger. I studied architecture there. Yeah, buildings and building technology, and I enjoyed it. But when I finished, I didn't like the architecture profession. I worked in an architecture firm for 2 weeks, and I decided that was not my thing. So I kind of went back to tech, and like 10 years after I first started with tech, I found myself back in tech. So it was kind of interesting. Then I started a new company with 2 other guys there, right there operating the live stream for the patients. We made an app called Ding Dong, and it was a location-based messenger app, and I was really excited about it. It was 5 years ago. I came to the Bay Area for the first time. I planned to go for a week, ended up staying 3 months, changing my ticket all the time. I liked it here. We ended up raising some capital at some point, and we moved to Berlin. So this is kind of where the story of my NF2 diagnosis starts. One morning in Berlin, I woke up and I felt something wrong with my left ear. Like everything sounded like glass. Like I flushed the toilet and it sounded like somebody threw over a porcelain cabinet. So that was really weird, and they didn't know what was going on. You know, I had like treatments like with steroids, and it went back like a little bit up and down. And then, meanwhile, we were working on a new product, and that kind of starting to take off. So I was kind of here raising money in Berlin, having my little hearing issue. So it was a very, very intense period. Because we didn't really know what was going on. So we moved, we were kind of in the process of moving here, and then they decided to make an MRI scan. And I remember walking in the doctor's office and seeing like a tumor on his computer screen. So I was like, fuck, a tumor, like, yeah, like that. They said they made the MRI scan to check if, you know, just in case there was a tumor, and yes, there was a tumor. But they told me it's benign, and you know, you will lose that ear, and we just cut it out at some point, and you know, you'll be fine. So I was like, okay, you know, no big deal. You just lose an ear, like it takes, you know, like you get used to that, like after a couple of months pass. Then we actually made the move here, and later we made another MRI scan to check, did that tumor grow or did it stay the same? And well, it turned out it grew, so okay, that was not nice. But then they also said like, oh, you also might have another tumor on your other side, like on the other hearing nerve. And you may have this disease called NF2. So I was like, okay, and they told me to Google it. And just beware when your doctor tells you to Google something you might have, because you see the worst cases like pop up on top, right? Those are the cases that go viral and are the most sticky. So I saw like people go deaf, they go blind, they get paralyzed, like end up in a wheelchair. It wasn't pretty. But they were not sure, so they made another scan that was like more precise, and a scan of the spine, and then they said, yeah, we confirmed it was a tumor, and you also have a tumor in your spine. Like, oh, okay, third tumor, you know. So, you know, it was kind of a roller coaster of bad news, until I talked to a doctor in LA, and he said, oh, you're 33, you only have 3 tumors, you have a mild case. So I was like, this is the first good news I got in a while. Like, on a parallel track, when I was here the first time, like 5 years ago, I met a few guys that were in genome sequencing, genome analysis. And to be honest, I was always fascinated by it. So every time I would go here, we would kind of have a little chat about what they were doing. I thought it was fascinating. And yeah, their software, they're making some software that actually studies the genome of one particular individual. That's how they got started. So when this happened, we were like, okay, let's try, let's do something. Let's sequence me and my tumor. So that was one of the reasons why I wanted to do the surgery rather like sooner than later, right? So I would have the tumor tissue, and I could sequence it, and maybe we find something useful. So as you do this, it's really important to do it from a point of like curiosity and exploration. Like, especially it's so personal, your friends are looking at it, right? You have to kind of have a mentality where you're like, I don't really care what comes out of it. Like, if nothing comes out, it's fine, we'll see. So we had a very explorative approach. I think on this end, we are in San Francisco now. We had a very explorative approach. So we did that, we sequenced it, and at some point he came to me and like, oh, we have some results, like there might be another gene affected too, they seem to be related, like they did some pathway studies, and there was a drug that would target that other gene that was affected. So for me, that was a big turning point, because I thought, hey, this actually seems to pay off. You know, it seems that what we had been doing was leading to new information. Is the people we talked to never really checked for an EGFR or a mutation in that other gene. Like, you know, most of the sequencing was done in the genetic testing in a gene in chromosome 22. But we'll get into the technicalities later. So they kind of got me started on thinking like, how can we do this also for other patients, and every patient we would probably do this analysis with would maybe help us understand the situation better on an individual basis, but also on the situation as a whole. So that's when I started thinking of NF2 projects. Yeah, so that's kind of the journey, what got me here. So I lost my hearing on the left side, and they cut out the vestibular nerve as well. So I had to kind of learn how to walk again, basically. Like, there are two friends of me here that took me from LA, from the hospital, drove me back to San Francisco. And yeah, I think we can all remember that it wasn't that easy to walk. But a couple months later, I was skiing, you know, doing a black diamond, whatever. So your brain kind of relearns how to deal with a new situation, which is quite impressive to see, like how your brain does that, which kind of gives confidence probably to anyone. Like, if you have a problem or you're losing something, yeah, you have a little bit of grieving, but ultimately you'll learn how to live with it, and it becomes the new standard. Yeah, like, the second part of something I want to share is around NF2 project itself. I mean, it's all very early, and it's just the beginning. I don't know if anything is going to come out of it, anything useful. So it's your typical project, right? But the same as we did the sequencing for me, we were doing it from a point of curiosity and exploration. I strongly feel there is something here, so putting all my effort in it. But yeah, it's also a path that is kind of unknown. So to me, that's an exciting thing. There are a bunch of things that I think that motivate me to do it. One is, I think the patients, it would be really nice if the patients play a more active role in medical research. So it sounds actually funny, but the patient is there, but it doesn't actually play like a crucial key role. It doesn't seem to be the user, the customer. You know, it's basically a pretty old system that feels, you know, ready to be challenged at least. So I'm really motivated when I talk to patients. I see them like they have so much energy to go after something and to kind of find a way to deal with it or to fix it. one of the problems is they don't necessarily know how. So I am very lucky to be among people that actually know a way to look at it, like, and amongst all of you. But most patients don't have that luxury, so that's for me is one of the reasons to do this. Another thing is that I see a lot of new technology coming up here, right? Like AI, the genomics becoming much more accessible, cloud computing. Those technologies, like, we are all very familiar with, or most of the people here are very familiar with it, but it doesn't mean that, you know, the rest of the world is. So I think creating a bridge and bringing all these new technologies to these problems, that is another motivation for me to do this. Yeah, so I kind of want to see if we can lay the groundwork for more pragmatic and actionable research. So a patient, you know, they just want to know which trial should I sign up for, what food should I eat, what food should I avoid. Is stress an important factor? Like they want to know something they can actually work with, and they are looking for that. And I think when you start with the patients and involve them, and they become the starting point of things, you know, we might actually automatically shift into more actionable research. Yeah, so like this is would be the diagram, but one of the things that really excites me about this event and how it came together is that there are definitely patients involved, and they are actually, a part of them are watching on the Facebook Live stream. So that's really cool as a starting point, but also there will be people from research. Marco is going to talk about it, more the biology field, and people from the computer world. And I think when these things come together, something really cool can happen. So the third thing is just about this event, the hackathon, right? We actually had a lot of fun organizing this event, and I want to, you know, before I say anything else, I want to thank the people that put their heart in it, because there's definitely, what I've seen is that the people that were coming to volunteer, there was much more for them to do this than only organizing another hackathon, right? Like, I mean, I'm just an example case, but anybody can get a problem like this, and a lot of other problems you might have in your life, like, become less relevant, let's say. So I think we all feel that when we were organizing this, and I'm really grateful that everybody that helped organizing put so much of their heart into this. So yeah, what do we hope to achieve? Well, I think we can't really know. I think this is probably a hard hackathon to participate in, because it's a really hard problem we are dealing with. And I think we need a lot of creativity and kind of boldness to look at this problem. Yeah, and I think it would be great if we have more questions at the end of this hackathon, or maybe we find even novel approaches. But what is it we need to look at next? That's going to be really interesting to know, apart from maybe other results that might come up. Yeah, and another thing I want to point out is we had some discussion around IP and ownership of data, and how open should this be. I think in a hackathon like this, I feel strongly we have to be as open as possible, right? So this hackathon is not about creating new IP. It's about getting to know each other, collaborating, and coming up with novel ideas that might actually help patients one day. So there was like a little bit of a journey. You know, we have like the data privacy we need to think about. Like, you know, I can, in this case, do whatever I want. I'm a person with the data, I'm sharing it with you guys, right? But later, it would be great if we can do more people and involve other patients in that might not even be in the room. So, yeah, it would be great if you all like can read this agreement and kind of, you just realize that it's important that we do this well to set a precedent for future hackathons to come. And yeah, I'm very happy to be a part of the experiment. That's all I have. I want to close with a little tile that I got right before the surgery. Actually, my mother reminded me of that nail clipper story, and she gave me this tile because I was already kind of talking about, hey, maybe we'll find something using this genome sequencing with the people that I know. So yeah, these problems could cause a lot of solutions. Who knows where it ends? That's where I want to leave it. I don't know if there are any questions. I'm happy to answer.
Are there any questions for Ono? Just one or two questions. Yeah.
Have you started in personalized therapeutic process?
The answer is not yet. We found a potential therapeutic, but again, you never know if it works, first of all, and there are like reasons to start with it now or start with it later, but you know, that would be the goal for me in the short term. Like, find an existing drug that's already on the market, FDA approved, maybe not labeled for NF2, but we can find something that I could like try out with relatively low side effects. Then in the future, like, we might come up with other things like the gene therapies, gene editing, and all these kind of things. But yeah, in the short term, we just are looking for an existing drug that's already like known and well studied.
Did your doctor give you a checklist of options?
The question is, did my doctor give a checklist of options? Not really. There are like different clinical trials for NF2 running right now. I don't know if Marco is going into. Marco arrived. That's nice. Thank you. Welcome. I don't know if Marco is going to talk about that, but there are like 20 drugs that have been trialed for NF2. None of them really made it through the whole cycle, and they all have pros and cons, and they work for some and they don't work for others. So that is something in the hackathon we can actually try to look at.
My last question: any one of these drugs involves immunotherapy?
So the question is if any of the drugs involve immunotherapy, and the answer is no, not yet. And the reason is, I mean, I think there is some work being done on that, but again, Marco probably knows more about that. Like, NF2 patients are not expected to die anytime soon, mostly, so you don't want to risk too much with your therapeutic. You know, so you have time to try a bunch of things like, without, you know, without dying. Yeah. Any more questions? Oh, there.
Hi, my name is Mary Grace. Just out of curiosity, where did you get your tumor sequence, and also did they sequence a specific tumor and on all three sides, or just your whole genome? And did they sequence normal tissue, surrounding normal tissue as well?
Everybody heard that question? Yeah. So the question is if where I got it sequenced and how. So we did it with the Broad Institute, or Broad, or however you want to call it. And all the sequences were done, the whole genome. And we sequenced my blood, the tumor that was removed on the left side, and my brother. So we did those three, and those are the 3 data sets you will have access to as well. What you had another sub question to that? Yeah, like I can actually share another small story. Like during actually Marco was there during the event. Someone asked me the same question, like, do you have another tumor sequence as well? Because it could really help. All these tumors can be different, right? So I was like, no, not really. I have another tumor on the right side and one in the spine, but you cannot really take a biopsy there, right? Then I remembered I had a little bulge here, and I was at a table with probably 5 pretty well-known NF2 doctors. And they were like feeling it, and they were like, yep, that's a schwannoma. And I was like, okay, great. Because this one can be easily removed and sequenced. So yeah, we may have another data set soon.
I have one last question. Can Marco use your laptop to present?
Absolutely.
Awesome. Yeah. All right. Great. So I guess a lot of the reason we're here is on the envelope. And I mean, in my personal opinion, I mean, having educated patients that are willing to share their data really leaps over a lot of regulatory hurdles. And so it's a really amazing thing. And in my personal opinion, Odo's really sort of a role model in terms of patients getting involved in their own treatment very, very actively. So that said, I'd like to introduce Marco, who is actually from UCLA. Not the Children's Tumor Foundation. But he did, in fact, fly his own plane up here, from what I hear. And he works on NF2 and is going to give us a clinical perspective on NF2. And oh, by the way, I have also been asked to announce, if you use Twitter, please use hashtag NF2hack. Yeah? Yeah, hashtag NF2hack. All right, with that.
Hello, everybody. I made it in time. Well, I'm Marco Giovannini. I'm a professor of head and neck surgery at UCLA, and I've been working on NF2 research for the last 20 years at least. And I had the chance and honor to be part of the team which identified the NF2 gene back in 1993. And then at the time I was a young postdoc. Then my next project was to develop mouse models of NF2. So mice where we reproduce the genetic mutations. The mouse has an NF2 gene which is almost identical to the human. So we just introduced mutations that are frequently found in patients and in tumor patients. And at the end, you know, after lots of crossing mice, etc., we were able to obtain mouse models of the 2 most frequent tumor types in NF2, schwannomas and meningiomas. And we have been using, we and others, we have distributed these mice to more than 150 labs around the world. And using those mice, we try to understand, on one hand, what are the function of the NF2 Merlin protein, and on the other hand, try to see whether we can use these models to test new therapeutic approaches. So I was very happy a couple of years ago when our mouse predicted that one drug called RAD001, or everolimus, a derivative of rapamycin, an inhibitor of the mTOR pathway, showed a positive signal in all the mouse models we tested. And based on that, and based on observation in an index patient treated in Paris by Dr. Calamaridis, we started 3 clinical trials, actually. And in the end, the results of the clinical trials showed that the mouse model was predictive of several outcomes, several observations we made in those patients. So that was a nice way to go back and forth from human to the mice and back. And hopefully this, we will be able to use these models in future trials. So this is new for me. The reason why I'm here is that I was very impressed. I mean, from the first time I met Onno, I thought, well, this guy is something different. Of course, I learned his personal story, but also, how smart he is, and the idea of being part of a community where, people would come in here thinking out of the box is extremely refreshing for me, and I wanted to be part of it. So when we spoke a week ago saying, I'm coming. But at the same time, I come with, my old school traditional background. And so I don't want to influence you in any way, in the sense that I don't want to filter, the creativity and the potential, any potential outcome of this reunion. So I was thinking that maybe my role would be to, the best way I could contribute to this event is by giving you clues on what is the disease and some basic fundamentals of the genetics involved in this disease. So I have no idea what your background is. You probably outsmart me in many fields, but so I tried, to give you an average level degree of information you might find boring, and of course we know it, or maybe you find something interesting. So let's see. So, and neurofibromatosis is part of a group of 3 diseases. Collectively, these are called neurofibromatosis. So neurofibromatosis type 1, that was called von Recklinghausen disease. And what is interesting is NF1 alone is the most frequent tumor predisposition in humans, the frequency being of 1 in 3,500. The second type is NF2, so 10 times less frequent, 1 in 33,000. And the last type, NF3, or familial schwannomatosis, is a kind of a newer definition of the disease. For a long time it has been assimilated for NF2, and only genetic analysis has shown that in fact it was due to different genes, but still the NF2 gene is involved, so it's kind of a hybrid form between NF2 and something else, which makes it extremely interesting. And NF2 as an individual clinical form was defined by Wishart in Scotland in 1822. It was also called central form of schwannomatosis because most of the tumors occur in the central nervous system, in contrast to NF1, where the periphery, the skin, and peripheral nerves are involved. So it's only in 1987 when the 3 entities were officially recognized as 3 different diseases, and that basically because of molecular studies, cytogenetic, chromosome analysis, and linkage studies that showed that the 3 genes were located on 3 different, actually on 2 different chromosomes. So chromosome 17 for NF1 and chromosome 22 for NF2 and schwannomatosis. You can interrupt at any time. This is not formal, not a formal presentation. So about 92% of individuals with NF2 have been shown to carry NF2 mutation, in the second or greater generation of family. Why is that? The reason is that about half of NF2 patients are sporadic cases, so they are new mutation. They don't have a family history. So it simply happens that the mutation occur during their embryonic development. And of course, the phenotype becomes fully penetrant in the following generation, because about 30% of that 50% of sporadic NF2 cases are mosaic, meaning not all cells in their body carry the mutation. So that gives rises to milder phenotypes, so maybe in segmental forms, patients that have only tumors in a leg or only in the vestibular nerve. And but of course, once the mutation is transmitted to the progeny, it becomes fully penetrant. What else? Yes, loss of expression of the NF2 protein, Merlin or schwannomin, has been shown in both sporadic and NF2-related schwannoma. Okay, so this is the first clue. Schwannomas are frequent also in the general population, not only in NF2 patients, not only in people who have a germline mutation of the NF2 gene. So any of us can develop a schwannoma. And together with meningiomas, which is the second more frequent tumor type in NF2, they are the most frequent tumors of the nervous system in humans. So also sporadic schwannomas carry biallelic inactivation of the NF2 gene. Another important point, we used to show this paragraph to the pharmaceutical companies, trying to convince them to invest on a rare disease, because although NF2 is relatively rare, we said 1 in 33,000, population studies showed that about 1 in 300 people, again, not NF2 patient, general population people, will develop a tumor in their lifetime with an underlying sporadic NF2 mutation. So pure random chance of getting an NF2 mutation, resulting in the same tumors that we see in NF2 patients, but also in other tumor types. For example, mesothelioma, which is a malignant tumors of the pleural, the membrane around the lungs, which is usually triggered by exposure, professional exposure to asbestos, carries a high frequency of NF2 mutation. About 60% of these tumors have NF2 mutations. But also kidney tumors, breast, melanoma, liver tumors can carry NF2 mutation, usually in addition to other gene alteration. So this is important because if we find a treatment or a new therapeutic option for NF2, that option could have much broader therapeutic application, and pharmaceutical companies like that. So most
would it be possible to use genome edition for this kind of disease?
To use what?
Genome edition, like CRISPR-Cas.
Oh, CRISPR.
Like yeah, gene
edition.
Gene therapy. Would it be possible to use gene therapy now that you have pinpoint the genes related?
Yes, so you are referring to gene replacement therapy in this case. Science fiction, but yes. In theory, yes. If you find a way to bring the CRISPR vector to the cells that carry the NF2 mutation. Of course, you must know the mutation in advance, but then you can engineer the vector. The vector goes in the cell, and the cell, the vector fixes, the mutated gene, and the protein is re-expressed, and thus everything goes back to normal. That's the concept. I don't think that anyone is there yet.
They're beginning just. They're beginning to do the test in humans this year.
But there are other genetic diseases where this, has been achieved. For example, in a rare form of child immunodeficiency, that was a team in France when I worked there, and they know them quite well. But of course, then they had roadblocks because they fixed the immunodeficiency, but then the kid got leukemia because the virus integrated somewhere else in the genome and screwed up another pathway. Okay, so I think this was first.
Have the NF1 and NF2 genes been replicated in mice?
Well, mice have their own orthologs. So there is a mouse NF2 gene, there is a mouse NF1 gene.
So is the sequence the same as human sequence?
The sequence is slightly different, but the amino acid sequence is almost identical. For NF2, it's that there is one amino acid difference.
So in that case, mutations are different between humans and mice compared to the normal gene sequence.
If you want to introduce a mutation in the genomic sequence,
in mice,
you can find a way. We found ways to mimic the human mutation in mice. I mean, you can approximate. Yeah. Okay. I don't know if I can hear you.
Maybe we'll hold discussions until a little bit later. Yeah, just there's a lot.
I'll be around all weekend, so. Unless you kick me out.
And these are great questions.
So, I already mentioned that mutations of both alleles of the NF2 gene have been found in tumors from NF2 patients, but also in patients with sporadic vestibular schwannoma. So those patients, general population, suddenly unilateral hearing loss, there is a tumor, that's what we mean for sporadic vestibular schwannoma. So both sporadic and NF2 carry inactivation of both genes, of both alleles. So just to give you, to summarize what is genetic mechanism, this is a classic, from a genetic perspective, this is the classic mechanism of inactivation of a tumor suppressor gene. Okay, so both copies of the gene must be inactivated in order to develop the tumorigenic potential of that mutation. So one allele is not sufficient. So in the case of NF2 patients, one copy is mutated in the germline. So every cell of the body carries one NF2 gene mutation in one chromosome. And what happens, the second copy, and usually it's a point mutation, then the second allele of the NF2 gene is mutated, this time usually by deletion, or can be another point mutation, but usually it's a more or less large chromosomal deletion, resulting in biallelic inactivation of the NF2 gene. So, if you look in the blood or in a fibroblast, in whatever cell of the body of an NF2 patient, you find one mutation. If you look in the tumor, you will find two independent mutations. Okay, so this is the classic tumor suppressor gene inheritance and mechanism of tumor development. In the case of the general population, so people who carry two wild-type normal NF2 alleles, both hits occur in the cell of origin of schwannoma or meningioma. So there is a normal Schwann cell, suddenly this normal Schwann cell loses a copy of the NF2 gene. And then, sometime later, the same cell loses the second copy and the tumor starts. Questions on this?
Any methylation process involved between the...
Rare. Rare.
Rare?
So, where methylation could be involved is in this event, right? In the second hit. So there was a study done in the UK by Dr. Evans, and they showed that in contrast to other diseases, mutation, so inactivation, the second copy of the gene by methylation of its promoter, reducing, expression or abolishing expression, is very rare in NF2.
So it's prevalent in X chromosomes, right? Methylation of the other.
Yeah, that's the way the X chromosome 1 is inactivated.
But on this one, it's rare.
It's rare, and this is chromosome 22, so it's not X-linked.
Okay.
But, yeah. So I'm just repeating, using a different slide, but the concept is the same. Okay, this is an MRI scan coronal view of an NF2 patient head. This patient had the 3 tumor types that are more frequently associated with neurofibromatosis type 2. Bilateral vestibular schwannomas, ependymoma, about 4 to 8% of NF2 patients also develop ependymomas, a kind of central nervous, kind of glioma. And meningioma, about 50% of NF2 patients develop meningioma. So, if we do genetic analysis of these tumors, we found that all tumors carry biallelic mutation or inactivation of the NF2 gene, so they have NF2 minus minus. But if we test a normal cell from this patient, only one NF2 gene is mutated. Okay, so the concept of germline mutation. So this is something controversial that I want to throw out there because I think it's highly relevant to what you guys are going to do. Okay, it's clear that, and also from our experiments in the mouse, there is a number of evidences that inactivation of the NF2 gene is necessary and sufficient to initiate tumor development. Necessary meaning it has to occur. Sufficient is doesn't need other gene alteration or genome modification or epigenetic modification to result in schwannoma initiation. However, and I'm not a mathematician, this group from Canada actually, but it was like Canada, US, and UK came out with this mathematic model suggesting that yes, the 2-hit hypothesis, so 2 copies, but a 3-hit model could also fit very well in the genetic of NF2. So this suggests that yes, you need biallelic inactivation of NF2, but you also need something else. But no one has ever continued, taking this avenue. But I thought this is highly relevant for what you guys are doing, and that third hit could be something different in every tumor, or could be something different in a family of genes that all work in a specific cell signaling pathway, for example. So that's.
We have a quick question over here.
Do we have any understanding, either structurally or genomically, why it is very common, but it is this spontaneous deletion is happening frequently in this NF2 gene?
No. So, if I understand the question is why this locus as sort of an intrinsic predisposition to mutation, right? No, but that's a general question. For the NF1 gene, it's a gigantic gene, so I guess just the chance of a mutation occurring there could explain some, but still it's too frequently mutating. Just to think about, the size. No, I mean, that's an open question, not just in NF2, for every, in tumor genetics. Why some gene loci are more vulnerable and predisposed to mutation or alterations than others. Yeah, that's an open field, definitely, yes.
Could the third hit be an EGFR mutation?
I've heard of one. I've heard of one. What is interesting about EGFR is that functional studies from the very beginning, after the gene was and the protein was identified and studied, showed that there is a link. So Merlin, the NF2 protein, is just downstream of the EGF receptor. So the EGF receptor is a transmembrane protein, and Merlin is some membrane protein. So there are studies showing that Merlin is downstream of GF receptor. It makes sense. Oh, and there is another. Yes, that was a very old study. Very old studies, we don't do those anymore. But if you know, when they used to do, what was it called? Chemical mutagenesis, like nitrosurea. So they gave, these agents that generate breaks and mutation, random mutation in the genome. So they used to give these drugs to mice, and many, actually rats, developed schwannomas of the trigeminal nerves. It was one of the most frequent tumors obtained using that. Using that strategy. So when they sequenced, or I don't know what they did, because we didn't have the power to do massive genome analysis. So I think it was probably a focused candidate gene kind of approach sequencing. They found that nitrosurea was inducing EGFR point mutations. So there is a link between EGFR mutations and schwannomagenesis. Usually, when you mutate EGFR, at least in rats, the tumors started as schwannomas. Oh, that's something I have to mention. Schwannomas are benign tumors, so I will come back to this, very important. The rat tumors quickly transformed, and they became MPNST, which are malignant schwannomas. So MPNST is malignant peripheral nerve sheath tumor, suggesting that that direct mutation of AGR would result in a much more aggressive proliferation than that induced by mutation of the NF2 gene. Because our hypothesis, we published this a few years ago in my lab, is that the role of Merlin is to intervenes in the recycling of receptors at the membrane. So brings and takes out receptors. So especially recycles, so takes out once they're done, they've done their job at the membrane. So in the absence of Merlin, these receptors tend to accumulate. So you have a kind of a upregulation of the EGF pathway without having that strong oncogenic dominant effect that we see in the rat. I don't know if this is clear.
Marco, one question. Just to maybe clarify this concept, which I think is very relevant for the audience, because can suggest something really new. So let's say that...
Oh, this is very old.
It's very old, but I mean, we can
simply no one has
have a fresh view on the disease. So let's say that the 3-hit model, the hypothesis would be that you still find some tissue which has 2 hits, which is not a tumor, right?
Possible.
When you say 2 hits, both member of the chromosomes are paired?
Yeah.
I mean,
well,
not
necessarily NF2 hits. It would
Okay.
be 2 hits in another gene, like what happens in schwannomatosis. Would be 2 hits in the SMART1 gene.
And then the third one is connected to the second allele of NF2.
Yes, to the first allele of NF2, and then the second allele of NF2 is mutated sporadically.
So let's say,
somatically.
How do you actually verify this? We need to take more samples from the guy. I mean,
the genome is from one clone, same clone of the cell. Then they compute genome. his complete genome sequence
is from the blood.
No,
so
is
from
it
the
derived
germline.
from one specific
No.
clone of cells?
No, from
Many
a tumor
times.
biopsy. Yes.
So you'll have a mix.
But these tumors are quite homogeneous. So it's a very uniform cell population. It's not like neurofibromas in NF1 when you have Schwann cells, fat cells, fibroblasts, mastocytes. Schwannomas are, and as are meningiomas, the other tumor type associated with NF2, are almost exclusively composed of Schwann cells or meningeal cells. Of course, some tumor vascular endothelial cells, here and there, but those are not mutated, so you can filter them out.
So part of my role here is to keep us on time, and I think it's really important. I know, some of these discussions are amazing, but what I would suggest is that Marco goes around to each of your teams and you can have that discussion with him as the weekend goes on, and that's really going to be incredible. So,
Prior slide?
What?
Prior slide, the slide before this one. Go to the previous
On links to the slides.
Oh, this one. Well, maybe we can download the paper.
Yeah,
right?
helps on the site.
Yeah, we can pop the paper up.
Everything that involves
No big deal.
Oh, genetic epidemiology. Yeah, we should be able to download it. We can PDF. Yeah. So what's going on? 2016, 2017, two papers came out on the genomic landscape of schwannoma. Of course, this is the fashion. You cannot, if you try to publish a paper saying, oh, I sequenced the human schwannomas, it's already not enough. So you have to say genomic landscape, meaning DNA, RNA, methylation profile, and more, whatever, just more. So basically, this is what happened, especially the first one. I think it was a fantastic marketing success. And the second one, European, Sweden, came out later, of course, lower in the journal. Sorry, I'm just drifting. Anyway, they showed that, basically nothing new in the sense that, of course, NF2 mutations are important for schwannoma, we knew it. And then they show mutations in other genes. And if you look, I don't know if you guys have experience in tumor, sequencing of tumors, but whatever tumor you sequence, there are ARID1 and 2 mutations. I don't know why. They found ARID1 and 2 mutations. The first paper also identified a chromosomal translocation.
Oh, is that 2217 translocation?
Oh, I don't remember.
Is it a 1722 translocation?
No. Nothing to do with
chromosome.
No, different chromosome, different genes. I don't know. Can you please look if you see it in this tumor? The second group didn't find it. What else? Well, maybe we can, I think I gave it to you on both PDFs.
We will put it on the site.
I think it's important. What is important is also, since these papers are published, these people, in theory, are supposed to distribute the data, the raw data. So I think that adds a great deal of power to any genomic analysis, just merging. Oh, one big difference is both papers were on sporadic schwannomas, not specifically on NF2 schwannomas.
Oh.
I threw just questions. Something that I always found extremely intriguing and intellectually challenging is why mutation in such an important gene as the NF2 gene result only in 3 tumor types. Okay, so what are the determinants of the tumor spectrum? Why the tumor spectrum in these patients is so narrow, so restricted? why all these tumors are benign tumors. So what is a benign tumor? A benign tumor is a tumor that does not invade the neighboring tissues, does not give metastasis, and does not spontaneously, most of the time, in the case of schwannomas, they never undergo malignant transformation unless irradiated. So there is a huge debate on the use or non-use of radiosurgery for an F2. Unfortunately, this is more and more frequent. Not in our clinic. And I can elaborate why we are against it. Meningiomas is a different story. 90% of meningiomas are grade 1, so benign tumors, but the other 10% are grade 2 and 3, so progressively more and more malignant, so atypical and anaplastic. Ependymomas are benign tumors, but since they grow in the spinal cord, of course, patients, eventually become paralyzed, and that's another therapeutic or clinical management challenge. But usually they respond well to surgery when it's well done. So are the mechanisms, the tumorigenic mechanisms initiated by NF2 loss tissue specific? Could be tissue specificity the key to explain that restricted tumor spectrum? That means there might be cell types where loss of NF2 has no effect. Either kills a cell or is an innocent bystander genetic mutation and the cell doesn't care. That's one explanation. I tend to think that this is the right, at least one of the factors influence that tumor spectrum is also because when we first generated mice, knockout mice, that carry only one allele, mutated allele of the NF2 gene. So genetically, they are identical to NF2 patient. So we followed the mice, for tumor development close to 2 years. Turns out that mice do not develop schwannomas, do not develop meningiomas, no ependymomas, but they develop osteosarcoma, fibrosarcoma, liver cancer, highly malignant tumors. So there is a clear, not only tissue specificity of tissue vulnerability to biallelic NF2 loss, but this vulnerability is not conserved across species. No one is working on this, unfortunately. And this is not just specific to NF2. If you look at most tumor suppressor genes, retinoblastoma, a rare tumor of the retina, and that's it. APC, colon tumors, Phony-Poll-Lindau, very restricted, or tuberous sclerosis, all at extremely narrow tumor spectrum. So we already mentioned the fact that study suggests that other genes or epigenetic modifications could be involved and could explain different behaviors of these tumors, because that's something we should keep in mind. These tumors in NF2 patients tends to be multiple. So patients have bilateral vestibular schwannomas, so schwannomas of the acoustic nerve, which is like 95% of the patients. But they can also have schwannomas of other cranial nerve, spinal nerves. And what we see is that, well, first, an important clinical observation that as soon as we make a diagnosis of NF2, And this could be any time between, I think I've seen a 3-year-old NF2 patient, but I also had an 85-year-old diagnosis of NF2. As soon as we do the first MRI, we analyze the tumor burden, saying, okay, this patient has bilateral vestibular schwannoma, trigeminal schwannoma, lower cranial schwannoma, hundreds of spinal nerve, spinal root schwannomas, 3 meningiomas. So the tumor burden will not change during time. So no new tumors will pop up during life. So that explains that these are developmental tumors, so they start very early in development. And what we see during progression is that they simply get larger, but not all of them. So some grow, some don't. Some grow and stop. Some never grow. So that's what makes the clinical management of NF2 extremely challenging. And because we sort of, the best therapy for NF2 is do nothing. So what we call the wait and follow or wait and see approach, meaning MRI scans every 6 months in the case of growing tumors, or every year in the case of no growing tumors. So, okay, NF2 mutation, probably other gene mutation that can modify the tumors that are initiated by NF2 loss. So those additional alterations could decide whether the tumor grows or stops, for example. And what are the therapeutic implications of those other genes alteration? It means that our targeted therapy must take care also of those mutations, or hitting the pathway, the main pathway that is deregulated by NF2 loss sufficient to have a therapeutic effect. Finally, all this could lead to personalized medicine, of course, individual treatment option. But in this case, it's not just individual as a person, it's individual tumor therapy, okay? Because, again, we assume that other events influence the growth, the expressivity of the disease as far as individual tumor response to NF2 loss. So maybe in the near future we will, make biopsies of these tumors and do a complete genomic or whatever omic you can think of analysis, trying to identify those molecular targets that eventually can be coupled to existing treatment, to FDA-approved drugs that have been shown, safe and effective for other tumors and could be repurposed for NF2 tumors. That's the hope. And our working model, definitely. I think that's it.
Wow. All right. Are there any questions for Marco from somebody who hasn't asked a question yet? And a lot of people had fantastic questions and really led in good directions.
Marco, we have some data from one patient and a sibling. Do you know of other databases available for NF2? You obviously must. Also, do you know of RNA databases, gene expression databases, which may allow us to look more into the pathways?
Well, I think those two papers that I showed, 2016 and 2017, that's where the data is. The question is how we get it. I don't have it. We requested it.
I'm sorry, so you requested it, but they didn't?
No answer.
So to the extent
That's quite usual. So we believe to put some pressure, eventually it will happen.
So to the extent you can share with us, could you give us a clue on, for the current drugs that are in the pipeline, what kind of NF2 biology is this trying to tackle?
What kind of?
what kind of intervention is it trying to tackle?
I mean, nothing fancy or out of the box. You know, it's a classic approach of taking drugs that are approved for other indications in oncology and say, oh, maybe NF2 deficient cells have upregulation of the mTOR pathway, so we use an mTOR inhibitor. And then you have different situations. You know, groups that are, you know, stronger in preclinical testing, try to go as far as they can using whatever model, in vitro or in vivo model they have. Other groups are just, okay, you know, try. But, you know, nothing, I mean, it really, yeah, the beginning, very frustrating.
All right, so I'm going to introduce our next speaker, and then I'm going to say a couple of things. That was a phenomenal talk. And actually, as an academic and somebody that works a lot with biomedical literature, I'm very excited about the next talk. Adita from Iris.ai is going to try to tell us how we can differentiate quality research papers from identify quality research papers. I will just go with the title of the talk as it is written. So we are running about 15 minutes behind, which is totally normal for being an hour into an academic symposium. However, we want to not be an hour behind because we want everybody to have close to 2 hours to be able to scope their projects. And scoping is so important for these types of things. So what I would ask is that everybody only ask one question per presentation. It's awesome that you guys are asking questions. The questions you are asking are absolutely substantive and on point, but please, one question per presentation from now on. Everybody good with that? Any objections? Awesome. And yes, you can ask questions on Slack if you have other questions.
My computer died, so give me a minute.
Okay.
Okay, there we go.
Once again, just to remind you, if you do use Twitter, hashtag NF2Hack, and yeah, you can put all kinds of stuff on Slack. And, you know, you can put stuff in the general channel, or we can even set up a questions channel. If people have just general questions, Marco can jump on there, or I can jump on there, a bunch of people can jump on there and answer questions. Or you guys can answer questions. Oh, by the way, while we're waiting, just so I have an idea, how many of you would consider yourselves bioinformaticians? That's amazing. So about 30% of you. How many would you say you're developers? Okay, almost. There's some overlap there, and other people do other things, which I look forward to hearing about over the next 2 days. So without further ado, Iris.
Awesome. Thank you. So I met Laura on a boat outside of Oslo in Norway just a few weeks ago. And she told me, I told her what my startup does, and she told me about this event here and said, if you happen to be in San Francisco, you should come and make your tool available for everyone over the weekend. And it so happened to be that I was going to be in San Francisco. Actually, I moved here this week, so that kind of worked out very well. And actually, this comes with a little bit of, I'm not going to be too personal here, but it comes with a little bit of, especially, I find what is being done here, especially kind of personally important. And I do have a sister that was born with a genetic mutation. She was born almost deaf and almost blind, and also, and it's called Rubinstein-Tybee, and part of that is also, you know, higher risk of growing tumors. So while it is very different, it is also something that is very close to my heart. So if what we're doing at IRIS can help any one of the teams during the weekend, then we are very, very happy about that. It is, I'll talk a couple of things about kind of quality research papers, but like a lot of you are researchers already, so you know that not all research papers are good research papers. But more importantly, so what our tool does is actually help find research. And we started this project about 18 months ago, and it stems from, so there's a number of reasons why we started this company. One of them being a previous startup I worked with where we had a big problem that we wanted to solve, and we had a team, and we had the starting point, and like surely someone out there, some smart researcher somewhere must have found information and knowledge that we can help solve this with. And I sat down in front of Google Scholar, and I'm not an academic and specifically not in that field. And I sat down and tried to, do a Google Scholar search. I'm like, I don't even have the vocabulary to try to find, like, I don't even know where to start, right? And that was kind of the problem that we set out to solve with Iris. And hopefully, for some of you, because you're cross-disciplinary teams, you might have a really good idea and think, could we be working in this direction? And what we hope is that our tool will be able to help you explore the research around around whatever problem that is that you're working on, a specific problem track, and that, and we have data on it. It actually works. So it's not just a hope. But we will be making our premium tool available for you over the weekend. Very quickly, kind of the 3 key problems in trying to map out the academic world around a problem that you're trying to solve. And I'm saying problem in general because you will all be working probably on different solutions to the same overall problem. There's 3 key problems. One is the keywords are really limiting. Right? So if you're not a deep domain expert on NF2, you don't know how vocabulary has changed over the past 10, 20, 100 years, right? So knowing some vocabulary of current, temporary sort, you don't actually know what words were used 10, 15, 20 years ago. So that's limiting. Finally, and also, of course, result lists, when you do, Google Scholar or PubMed or other searches, you end up with these massive result lists that take a lot of time to go through one by one. And of course, the citation system, we can have lengthy discussions about that, but less than 15% of citations are actually related to the problem that you're trying to solve. So while it does have merit, sitting and looking through all of those citations and doing the kind of snowballing to find relevant research papers is a hassle. So that's what we're solving with our tool. Very quickly, and I'll give you a demo, and I'll focus more on that rather than giving you the pitch. But we use a combination of natural language understanding combined with a pretty different UX than most of you are going to be used to map out research around a problem statement. So I'm going to give you a demo, and I'm also going to I'm going to post on Slack. I'll send it to Ono as well, so you can have it if anyone is not on Slack. I'll give you kind of the login to the tool, the problem statements, all of that. You'll get the slides as well, so you'll have all of the information. And I'll give you a demo now. Let's see. So the tool that you will be given access to starts with, and very briefly what we do is that we work with a full text problem statement. So you will be given a problem statement that is a little bit general around the different problem tracks. You can start there. You can write your own text, copy from Wikipedia if you find a specific thing, if you want to dig into specifically around tumor growth, for example, or whatever it is. You can pick a full text problem statement to describe the topic that you want to dig into. What we do is that we work with a number of different algorithms to create what we call a fingerprint of this problem statement that you're trying to solve. We work with the words of the text itself. We work with contextual synonyms. We work with topic modeling to really map out the full context of what you're trying to find solutions to. And then we use that to do fingerprint matching, essentially, with other research papers. And that's how we work with this. So how it works is that you start with, as I said, a problem statement. I don't know if you probably can't see this in the back, but you will get access to it, of course. So you start with an overall question and a detailed natural language description of the problem that you're trying to solve. You give that to the tool to read, and then what we do, oops, I'll show you without the supervised search first. And then what we do is that we map out, these are the main topics that you're going to be looking at. These are the main topics of the text you inputted. And in this case, it's one of the questions that was asked. And you can dig in. So there's going to be words, because the system isn't perfect, there's going to be words that are not related. That just means you can skip all those papers, right? Because, and we'll get to the papers in a second. And that's the beauty of the UX of this. So let's say you really want to dig into tumors and growth in tumors, for example. And at the bottom layer here, you're going to find research papers. And we're drawing on an open access database of about 77 million papers. So it's a good starting point to start your search. And for example, I bumped into this one, for example, which is about mathematical modeling of tumor growth, which may or may not be relevant, right? But it's just to give you an example. When you so you can read the abstract. You can click to download the full text link. Most of these are, as I said, open access. You'll be able to download them and read them. We also have a bookmark feature, and actually the tool that you're getting is on our dev server. There is work that has been very recently done. Some of these features are brand new, and you'll actually be some of the first ones to try them out over the weekend. Love feedback on it. So to take this one as an example. We have this. So when you go through this, you start with your problem statement, and you start seeing the different topics, and you navigate your way around. When you find something that is interesting, you can bookmark it. And the way you do that is you actually connect to a Excel template that I'll send you as well. Make a copy of that. Authorize the tool to connect to that version of the tool, and you can start filling out this template and very easily bookmark the papers and add them and add a list. So you kind of create a report for yourself on the topic that you're exploring. And again, I'll send you the descriptions of this, but just to give you a heads up on that. We do have another, so, you know, in some cases, the tool isn't perfect, right? And sometimes, I mean, we make assumptions on the way. One feature that actually, and I talked to my dev team today, one feature hopefully will be connected by tomorrow morning, but I can't make any final promises. You guys know how it is to do development. But one of the features that we do want to, that we have, it is here in the front end already, and we just need to connect the back end to it, is what we call the supervised search, or where you input the full text, and what you actually get is an overview over the topics that we've found. That is part of this search or this navigation, and you're actually able to change this yourself. So you can, when you get you know, one of the main topics is neurofibromatosis, of course, and you can jump into it, find a definition, and find potential synonyms. If there's one of the words you think doesn't make sense, go in and find a synonym and replace that. You can also drag and drop and move things around. So you can actually change the hierarchy a bit and rearrange the papers so that you find exactly those papers you want in that bottom layer. Does this make sense? This is the first time I'm explaining this to someone, because it's the first time we're demoing it. So you can move this around. Right now, if you try this tonight and you know, work with this supervised organization of all the papers, you're going to find a demo setup with demo data. But as far as I'm aware, with 90% chance, you'll be able to see the final results tomorrow morning in whatever problem that you're trying to solve. So that's that. A couple of things. Any questions about the tool? This is a very quick demo, but any questions about the tool? I have a couple more slides after that, but.
Are you computing like a low-dimensional document vector from that input field and then doing like a nearest neighbor type of thing to find the papers?
I'll show you what we do here. So I can send you the paper on the document matching as well. So what we do is there's a number of different algorithms. So we do keyword extraction. TF-IDF is a statistical model. Sounds like you know a little bit about these things. So we do first keyword extraction, then we work with contextual synonyms. We do word embeddings. We train the model on 5 million research papers. This is all cross-disciplinary, so it's definitely not just genomics. You're going to find a variety of different fields. So whatever field you want to dig into, as long as it's on the harder sciences side, we're going to support that. So we do contextual synonyms by training the machine to find contextual synonyms using word embeddings. And then we do something called neural topic modeling, where the machine clusters all of the research papers, about 5 million research papers, cluster them based on hidden topics, and then we use a word embeddings model to put labels on those topics. And then all of that is used to form a fingerprint. There's a list of words, but it's like each of the words are weighted based on their relative importance to the original document. And then we have a similarity metric that we use to match that fingerprint to the fingerprints of a number of other documents. So that's approximately what we do. And then we do create these topic hierarchies, and that's a heuristic function. It is not always perfect, hence the supervised place where you can actually go in and drag and drop and make your own hierarchy and distribute the papers into that. Yep.
Cool.
Right. Let me find my screen again. Right. Any other questions?
While you're bringing it back, can you talk about how you deal with abbreviation expansion?
In terms of, yeah. So, so we work with, I mean, because we work with contextual synonyms, we find a variety of different names for the same things. So, I will say one thing, because we got the problem statements, a few days ago, and started testing it, and we found a bug, because we're not perfect. So, we don't do, what's the word in English? I have it on my slide, if I can get it back up. Basically, numbers and letters in combination to form a word, which is very convenient today because we're dealing with a disease called NF2. But we can use the full name of the disease, and you will find it. So that was a little bit unfortunate. I've been working with my dev team the past 24 hours to try to get that fixed on, but unfortunately, that's not going to happen. But we do, because we work with the contextual synonyms, you will find abbreviation and full text of the full versions of the words, but not with NF2. Yeah, alphanumerical words was the word I was looking for. Very briefly, because we were in a room full of scientists, our tool looks different, and there's some cool tech in the background, but it also actually works. And we've tested this, and this is also one of the reasons why we're invited. One of our ways to prove that the tool works is actually to do hackathons, what we call sci-thons, science hackathons, very similar to this event, where we form small teams of multidisciplinary researchers to solve a variety of challenges, and then we measure the results with an external expert panel. So we have proven that using this tool, if you use it consistently in your research landscape mapping process, actually yields far better results than what you do if you sit with, say, Google Scholar, try to find your own keywords, try to formulate what you're looking for in keywords on your own. This one, and I'll send you the slides, but this one experiment had teams that were not domain experts whatsoever. They were engineering students, PhD students. In this case, the team that used our tool more than twice outperformed the teams using Google Scholar, and that was on key papers found, on the overall structure of their findings, and on the conclusion they were able to draw after about 4 or 5 hours of dedicated work. Another example was that, and we did a little bit of a broader example, where we had 7 teams that, again, they sat for 7 hours, tried to answer a question around lifestyle and non-communicable diseases and lifestyle changes. And again, they were able to, they could choose whether or not they wanted to use the tool. And what we saw that basically the more you use the tool and the more consistently you use the tool in that early mapping out of what is the research world saying about this, the better result it yielded. So that's, of course, very exciting for us, and hopefully proof enough that this will provide you value if you use it over the weekend for those teams who will actually be doing research, of course. There's a question there.
Yeah, sorry, one more time. Has your product been validated by research labs or universities?
So we have, this one was actually done with Uniclinic Freiburg. There's a group of researchers. They do urology. So, but the process and the teams were from the Uniclinic Freiburg and doing that. So they were part of the whole process from start to end. We also have one peer-reviewed paper, but on a small part of the algorithm. I don't know what's up with my computer today.
Does the result also includes the non-open source competition as well?
For now, yes. I mean, we do, when we work with kind of enterprise clients, we are able to connect their credentials to the tool, but that's not set up on a ongoing basis. So for this weekend, yes, it's open access only. So it's from the core project in the UK, so you have about 77 million articles.
How does the quality of the search results depend on how much or what you put in the search box?
Right, so suggestion is 300 to 500 words. It has to absolutely be 100. Any less than that, and we have no context whatsoever, and you might as well go out and do a keyword search. So 300 to 500 words is the optimal. And then it depends a little bit how broadly you go as well. You'll quickly get a feeling for kind of how the results, how the results are yielded, depending on it. But 300 to 500 words. The thing is, you can use your own words in this, right? You don't have to be very, very specific in terms of exactly what words you use, because we deal with the synonyms and the topic-related words as well.
Great. Well, I think I know what I'm going to be doing for much of the weekend, which is playing with this tool. So that should be a lot of fun. Do you have more slides?
I think it's fine because we're running a little short on time. But I will not be here anymore tonight. I will be taking off, but I'll drop by tomorrow, like around probably when is it open here? 10? Yeah, 10 or so. So if anyone has any questions or want to dig into the tool or ask more or find a bug or can't get it to work, I'll be here for at least an hour or two for those who have questions. So happy to help there. And I'll send all of the details and information and login and things like that on Slack.
Great. Okay, well, thank you very much. All right, our next speaker is Burton Earnshaw from Recursion Pharmaceuticals, and he's the one speaker I haven't actually met yet. Hi. Hi, Bert.
We're gonna fit Marth in real quick before Bert.
Oh, okay. We're popping in another speaker before Bert. Here, do you have slides? Pretty good.
Nope, I'm just going to talk. Hi, so I'm the developer of a library called DeepChem. It's a deep learning library for drug discovery. I hope some of you will use this library this week to propose new molecules that could potentially be useful for NF2. So the process for using libraries like DeepChem. A little louder? All right. So I'll just kind of repeat the blurb in case people didn't hear. I do DeepChem. It's a library for using deep learning for drug discovery. I hope you'll use DeepChem to potentially find new molecules which can help with NF2. The key thing is you need to, I'll say 2 sentences, come ask me later. Find protein targets potentially associated with NF2. Find public data off databases like PubChem or MoleculeNet, Google them, and use DeepChem and graph convolutional networks to build a model. Now, those are very concise sentences. Find me if you actually want to know how to do this in practice. And if you want to write a tutorial, you will be immortally remembered in the docs. Thank you.
That was phenomenal, and thank you for the PubChem.
Bart's is going to do a DeepChem tutorial tomorrow morning for those of you who like to join. We'll post the details in the Slack. For those of you that are interested in the DeepChem problem track for the weekend, Barth is leading that. And is Patrick here? Patrick here? And Patrick too.
All right, awesome. I am looking forward to seeing that project. That should be very exciting. All right, so next is Burton Earnshaw, who I just met. He has a hell of a handshake, so regardless, you should shake his hand at some point. And he is going to be presenting in just a second.
I'm being forced to make a PDF.
Oh yeah, he is making a PDF right now. But isn't it presenting? Can you just present from there?
One second.
All right. Anyway, it's going. What was I going to ask you guys? Yeah. Oh, great. Cool. How many of you have any experience with natural language processing? Out of curiosity. Wow. Like that is kind of amazing that so many of you have experience with NLP. That's great. All right. Without further ado. Here's Bert.
Okay, great. Thank you. Let's see here. All right. Well, thanks to the organizers of this hackathon. This is a pretty amazing event. I've been really inspired just to kind of hear how this came together and to hear Ono's story and just really excited to represent Recursion Pharmaceuticals, a sponsor here of this event. I am the director of data science research at Recursion. We are a drug discovery company, but very different from what you might think of when you think of pharmaceuticals. And I'm going to take a few minutes here to tell you a little bit about that. So, drug discovery in the year 2017 has really a lot lacking. This is something I've seen a lot since joining Recursion. That's where it's getting cut off. Well, this is the number of drugs created and so developed and approved per $1 billion spent over time since 1950. And you can see this is going down and to the right. This means that there's less and less drugs actually being created, or said differently, there's more and more money being spent to create less and less drugs. And there's a whole lot of reasons for that. I will talk a little bit about kind of how typical pharma goes about this, and then how Recursion is very different. What it does mean is that there's lots and lots of diseases that go kind of unlooked at. We're talking about NF2 here, and Marco mentioned how he'd love to see pharma take us on, but it's a rare disease, so nope. This is in our commons room at Recursion. Our headquarters are in Salt Lake City, Utah. That's where I'm coming from today. And these are patients. Notice that they're all children. So about half of patients with rare diseases are children, and these are all children that are, in some ways, we're associated with their families. It's happened through different means, but we're really inspired by their stories, and really would love to be instrumental in helping cure the variety of diseases that are out there that are typically gone unlooked at. They go, well, for lack of time, I'll kind of just go through this. In contrast to where we are with drug development and discovery in 2017, technology, of course, has made like enormous strides, right? So this is a part of our lab at Recursion. I'm not a biologist, but what you have here is like a seriously awesome and fairly expensive automated lab for running hundreds of thousands of experiments every week. I'll go into some detail on that. So we have like amazing instruments from which we can do lots of great experiments. Of course, we've got access to all the computation we could possibly want these days, as long as we want to spend just a little bit of money. And AI, deep learning, machine learning, is just made such strides in the last 10 years and even the last couple of years, and it's so exciting to watch the field move. It's hard to keep up with it. There's just so many things that we didn't realize we could do, or we never thought we could do maybe 10 years ago, that we're doing, and even more, in particular with deep learning. So what's holding back progress on drug discovery? Well, here's the standard formula for a pharmaceutical company. You pick a disease, and then you do all kinds of research, spending all kinds of money, trying to come up with hypotheses on the biological compound that might help this particular disease. You do very, very low throughput single endpoint experiments, trying to validate that, and then you're making a monstrous bet on a couple, just one or two candidates. I mean, at this point, you spent billions of dollars, and that bet sometimes turns into ill-advised drug delivery. This is not a good way to go about things. It's very slow. It's very it doesn't cover the space of drugs and diseases. Can we do better? Yeah, we can do a lot better. So this is something that I'm looking at every single day now. This is a high-resolution image of various channels. I'm going to go through each of the channels here individually here in just a second, but this is a composite picture of human cells. And it's got a lot of information in it. So there's various channels here. What we do, our basic experiment is to treat cells with various fluorescing probes that will bind to different parts of the cells. So here's a probe that's binding to DNA. There's another to the endoplasmic reticulum, to mitochondria, Golgi apparatus, cell membranes, RNA, nucleoli, and actin fibers, structural fibers. And when combined, you get this picture of what the cells look like. And it's really high resolution here. I mean, we can keep digging in, and we see all this detail about what's going on in these cells. And this is an image taken from one of the hundreds of thousands of experiments that we're doing every single week. What's great about images is that relatively they're cheap. I mean, once you've got a lab, then they're cheap to run week after week after week. We run these in parallel and literally doing, hundreds of thousands of experiments. What are these experiments? They're typically they're taking a cell line. We have there's various cell lines to be looked at. We perturb them in some way. We are very focused at the moment on monogenic loss of function diseases, many of which end up being rare diseases. And we have a disease model here in these cells. We can look at see what those cells look like when they're diseased, and also what they look like when we apply a drug to them. So can we fix drug discovery? Yeah, we think so. So what you see here is a healthy child and a child with a rare genetic disease, Cornelia-de-Lange syndrome, and we're pretty good at just looking at the phenotype or what these two different children look like, and we can say, yeah, we can make this diagnosis. Of course, not all the diseases are like that, but we want to do exactly the same thing just with cells. We want to be able to look at cells from healthy or wild-type cells and those that have been genetically perturbed through something like gene knockdown. And then we want to apply drugs to them. And we're not just looking at a single drug over 10 years. We are applying basically every drug we can get our hands on. That would be off-patent drugs, off the shelf, over-the-counter type things, and also partnerships we have with large pharmaceutical companies who would like to try screening their drugs for repurposing. We want, the task we have here is to say, okay, when the drug has been applied to the disease, has it helped? Has it rescued this disease cells and come back to a phenotype that looks more like healthy? So how do we tell the difference? Well, this is San Francisco. This is an AI hackathon. We use data science, and there's a lot of things that we do here. As you can imagine, we've got multi-channel images. These aren't RGB images like you would see in a typical kind of neural network context, but we nonetheless have multi-channel images, and we can apply the kinds of neural networks that you would think to use here, like convolutional neural networks, and produce representations that live in a much lower dimensional space, and that should be very informative. And we use those to do a variety of things to solve to give us a space in which to ask the questions: did the drug rescue the diseased cells? We use it for doing other things too, like if you do enough experiments in the wet lab, you'll know that week to week, just things change, and so we can use more complicated networks, like in this case the striplet network, for sort of dealing with the variety and the changes that we observe through different experimental setups. Now, data science, our job there is to sort of fill the pipeline up with candidates of drug and disease pairs. Once we identify candidates with high enough confidence, this moves on to in vitro and in vivo validation. And then, of course, that moves through the clinical pipeline. And I'm showing a picture here, slightly outdated, but of 20 hits that we have identified through this process. You'll see actually in the in vitro section that NF2 shows up there as the second program. Again, all these are monogenic loss of function diseases. They're all rare diseases, I believe, and this is 20 programs. We've only been at this for a little while. I don't know that these will all turn into actual drugs that we will find will actually make it through the entire clinical process, but we have way more candidates in 1 year than any pharmaceutical company has ever done. And that's because, again, we're not designing and manufacturing these compounds. We are just taking what's out there and using them. So we don't use the standard formula. We're looking at hundreds, even thousands of diseases. This doesn't take 10 years. This is just, we're scanning these over months of time. We don't need a biological hypothesis. We're just letting the data tell us where the hits are. We're looking at thousands of different features of the cells in parallel, and we're going after hundreds of candidates. What's the 10-year outlook look like? We think of treatments in this way, as a matrix of diseases and drugs. We want to be able to basically scan this entire space. You might call it brute force search, but that's what we're doing. Currently, our approach is to just cover the space with disease and drug pairs and see where we might have some hits. We can and do incorporate information beyond just the images, what we know about diseases, what we know about drugs and compounds, and we can use machine learning and go after these things and find where the hits are. And as we do that, we learn models that allow us to actually go outside of this space and dream up compounds that don't exist yet that might target existing diseases or possibly diseases that we may see in the future. So anyways, hundreds of millions of images. Recursion has got experts in a variety of fields. We're using the latest in machine learning, deep learning, and our goal is to treat 100 diseases in the next 10 years. And really the main goal overall is to solve human cellular biology in the process of doing so. But that's what we're after at Recursion. Again, very happy to be a sponsor and be associated with this event, and really inspired by it. And, thanks for the time, and would love to talk to each and every one of you who would like to talk as well. So, thank you.
Wow, I mean, that's something I certainly lose sleep over, is how are we going to get enough drugs for all the monogenic disease. So thank you for trying to work at this problem. Any questions? I think we have a couple.
I'm just curious, when you're comparing images, are you looking at single channels, or are you looking at a composite image?
We're looking at the composite in the sense that our image is a tensor of, one 2D image per each channel. And I was showing 5 there. Our current experimental protocol has 6. I think the future protocols will actually have 8 channels of information. So lots of information there. Again, targeting the variety of entities and bodies within cells and the structure around cells. Yeah.
Will we be able to see your images of the NF2 cells?
No. Actually, we did ask about that, but the answer is no. I'm sorry.
All right, one more question.
In terms of your drug selection, are you using well-known combos like Avastin and Tarceva or something like that, or is it just some single drugs that you use?
So our typical drug screens will screen, on the orders of 1,000 drugs at a time. So these would be drugs that are no longer, they're off patent. So they have been patented in the past for a particular purpose, but as we're looking for another purpose, we can screen those drugs. We also do screen a lot of on-patent drugs that are our pharmaceutical partners' drugs, again for repurposing.
But how about combinations?
Yeah, we are not currently doing combinations of drugs, but certainly that's the next step. But we'll do the first order problem, then go to the second order problem.
Well, thank you again. That's awesome. Somebody who I haven't thanked yet is Sean. So Sean is working as our sysadmin here, which is awesome. He's also doing, obviously, a lot of AV stuff. And we are using his computer for the next couple of talks. So what we're going to try to do... Oh, by the way, how many of you have ever been to a hackathon before? Okay, so about half of you, maybe 2/3 of you. So 2/3 of you are not bioinformaticians, and a third of you haven't been to a hackathon. And so what I'm going to try to do is, so after I'm done, Carlos is going to come up and speak, and he's going to talk about the problem tracks and help you guys get into teams. And he is going to do a whole bunch of amazing things. So what I'm going to try to do before Carlos comes up, Sean, how do I get back to the PowerPoints? My PowerPoint. Awesome. So what I'm going to try to do is I'm going to try to give you a 5-minute introduction to bioinformatics and a 5-minute introduction to hackathons. So I will try to do that very, very quickly. Obviously, you may have some questions. I'll try to go through them quickly as well. But I'd love to talk to each and every single one of you here during the hackathon. I will be around the entire time to help out with questions. So I work for a place called SVAI. We house a lot of the world's biomedical data in a database called PubMed. We're also one of the largest genomics databases in the world. We share a lot of data with the Europeans and the Japanese. We have about 10 petabases of raw genomic data that comes out of sequencers. We also have health resources such as ClinVar, dbGaP, and GTR, which I'm going to talk about a little bit later. All right, so 3.5 ways of literature. Actually, there's 4.5 because you can always use iris.ai. But if you want to go to the raw databases, you can go to PubMed. You can search on the web. You can also get free full-text articles using PMC. Additionally, we have a command line API called eDirect. eDirect is nice because it also comes with an NCBI native XML parser. For those of you who are bioinformaticians, this may be useful. Just Google eDirect Cookbook. Also now, PubMed is available for download and is updated every day. And there's an open access subset to PMC. Just Google those things. And I will put these slides on one of the hackathon channels. We also have an alpha, an automated tool to work with NLP products if you're interested in that, which apparently half of you are. So that's awesome. The next thing I want to talk about are genes and proteins. So if I look at NF2, I can see that here we have lots and lots of different isoforms. And this, I think, is really relevant because if you look at the fourth one down here, I know it's a little bit hard to see, but you see that it's missing a lot of exons. So the thick lines are exons, the thin lines are introns, and if you're missing a lot of exons, you're going to get a totally different protein, and that's really important. And different isoforms are going to be in different proportions in different cell types. How do I know that? Well, for one thing, I can scroll down and look at aggregated RNA-seq, and here I can see various read counts of these things in a bunch of aggregated tissues. If I want, I can split out those tissues and look at them. The other thing I can see here, and I think this is so interesting, is in ClinVar. So what ClinVar is it is a database where clinical genetics labs assert variants that are associated with particular phenotypes. And up here, these variants in purple, these are variants in NF2 that are associated with various diseases. So you can dig into those. And also, it's worth noting that the ACMG recommends that if you accidentally find a mutation in NF2, you should report it to the patient, the American College of Medical Genetics, and we can talk about that later. So say you have this professional guideline, you can look at what these variants are, you can filter them by pathogenicity, significance, and also how many different clinical labs found them. So there's a crowdsourcing model there. But one thing that you may be interested in, and this is something I think about a lot, say you find a new mutation we don't know a lot about, and you think this may be associated with a particular rare disease, a nice thing you can do is look in the 1,000 Genomes Project to see how frequent it is over different human populations. So what's the 1,000 Genomes Project? Well, what we did was we took 100 individuals in 26 ethnic groups, so 2,600 individuals, and we did a whole lot, we calculated a whole lot of SNPs, and those frequencies can be seen in this genome browser and also downloaded it en masse. By the way, ClinVar, you can just download the whole thing as a VCF file. If you don't know what a VCF file is, don't worry about it. Going to the clinic, we have something known as the genetic testing registry, so clinicians can actually order tests off of some of these that are developed off of some of these particular variants. And we have a whole bunch of APIs. The last thing I want to talk about in terms of bioinformatics is next generation sequencing. So what we're doing these days, although the ends are a little bit longer, now somewhere on the order of 200, 225. So we have two ends, usually they're paired by about a 400 base pair linker, and they're all stacked up. And we map them to genomes. So this is a basic schematic of next generation sequencing. I'm going to skip over it. If you really want to take an online workshop, we put one together. It's about 6 hours long. It's not that bad. I mean, it's all of next generation sequencing in 6 hours. Plus, you can actually go on. I think we have it set up for Amazon and Google Cloud, where you can go and learn how to do RNA-seq mapping yourself. So if you're interested in that, check it out.
Audience for that?
What's that?
Who's the target audience for that 6 hour?
Who's the target audience? Basically, people that are just getting into bioinformatics. It's quite simple. The idea is that you have to have just the most basic experience with a Linux command line. But I mean, nothing complicated. Anyway, we have this new human genome resources page. You can download all kinds of stuff, including the VCF of ClinVar, the VCF of dbSNP, and unfortunately I don't have time to go into it, but also a database of structural variants. Those are variants that are more than 50 nucleotides called dbVar. And actually that may be something very interesting that potentially comes out of this. I don't know how much we've gone into the structural variants that are in Ono's tumor, but that might be another interesting thing. Also, we can look for public data sets. So here's a couple that are specifically in NF2, but after hearing Margot's talk, I thought a lot about, well, no, there's a lot of other things we should be searching for in terms of schwannomas, both in bioproject and the SRA database. So this is where we keep both the public and, in some cases, the personally identifiable next generation sequencing. And so you can go in here and actually get the raw data, and you can look at other mutations of microRNA and expression for a whole bunch of different diseases that may be useful to you in some of your products. So I want to say one thing real quick. Many of you know what BLAST is. If you're bioinformaticians, you certainly do. We have a new kind of BLAST. It works really, really well with next generation sequencing. And the nice thing is it's about as fast as HiSet, but you can make databases on the fly. So you can do things like call variants, look at expression for particular genes right away, and I'd be happy to walk you through that. You can go to the NCBI Learn page for more information on that. Here's some other bioinformatics resources. BioStars is great. Omics Tools is great. There's many, many, many other great resources. I want to talk for just 2 minutes about hackathons before I turn the floor over to Carlos. The way that Pete found me is a bigger and bigger part of my job is running genomics hackathons. So we've run 12 in the last 2 years, and we're going to run 12 this year, I think. One of the things we built lately is FenVar. You put in an RSID and get out a word cloud. Word clouds are not science, but you can see a network of those things mixed up, so then you can analyze pleiotropic SNPs. We're also interested in warning clinicians about reporting SNPs back to their patients when they're in regions of the genome that are intrinsically unstable, i.e. they have a lot of copy number variants. We do some immunogenicity things, and we write pipelines to keep things in RAM. Some of these pipelines may be useful to you, and I'm going to try to come around and talk to every single team, and I'll give you suggestions about these tools, as well as actual production open source tools that are out there. Right, so one of the most important things is that in our hackathons, we put everything on GitHub. A lot of them are in the NCBI-Hackathons GitHub site. You can check that out if you want. And we have upcoming hackathons, so here are a bunch that we've already set in stone over the next year. I would point out for those of you in the Bay Area, we're planning to have one the first week in April at UCSC, which also, if you're a bioinformatician, you know, is sort of an amazing sort of political thing as well. I thought of a couple other projects. I'm really interested in biological networks, and Marco alluded to this maybe 10 times during his talk. If we can look at biological networks that are affected, so say you have EGFR and NF2 and a bunch of other things in a network, and say we know of some drug that might affect that network, perhaps all of a sudden we have a new good topic. Also, can we make it simple to cluster subpopulations? Obviously, we have a patient of one, so that's not super simple clustering, but it would be nice to be able to do clustering and point them to appropriate clinical trials, which is something Ono mentioned as well. Finally, Pete has set up an SVAI GitHub repo for this. And what I would love to see, and what I think, what Ono would love to see, and what I think everybody would love to see, is even if you have a pure research project, even if you're not building any new tools, but you're running some scripts or whatever it is you're doing, set up a GitHub repo in this organization and document what you did so other people can see it. And other people can try to reproduce the thing, right? Because, I mean, there's lots of other rare diseases that whatever your pipeline is, whatever you're doing, whatever research you're doing, might be applicable to. So it'd be great to throw this out there. And one last thing I wanted to say about hackathons. So obviously we run a ton of them. I ran one at the New York Genome Center earlier this week, Monday through Wednesday. Obviously I'm from the East Coast of the United States because I talk insanely fast. But anyway, so one thing I would say is also I find that when we do things that are clinically related, it is awesome to have a senior clinician to bounce the ideas that you've thought of off of. And Marco will be here all weekend. So bounce ideas. Say, would this be useful to you? You know, I mean, that to me, I think really works well. The other thing I would encourage you to think about is scoping. Before you start writing code, I think projects are way more successful when people carefully scope their projects and really think them through, just like you would if you were doing a software development project at work. Try to scope it out. They typically go better. And I think with that, those are sort of 2 tips and tricks I have. I'm going to try to help you guys out with bioinformatics throughout the hackathon. And once again, if you're a bioinformatician, raise your hand. So any of those people can help you out with bioinformatics, right? Like, so yeah, just ask them too. Maybe I'll defer to some of them at some point. And you can also ask Carlos. Carlos thought of a lot of the problem tracks, from my understanding, and now he is going to walk through them with you. So.
I'm going to go over here.
Yeah.
Does that work? Yeah, that's perfect. Hi, everybody. So it's a little late. I hope everybody's surviving well.
Doing good.
A couple sleepy eyes, right? But thank God I'm following you. You have some energy. And that's a good thing. Because we do need energy to be able to, accomplish what we're trying to do here. So we're trying to find novel ways of looking at NF2 in particular. It's kind of spearheading this big approach. I'm going to talk about, I guess, 10 minutes, and just I want to touch 3 minutes on who I am a little bit, and then how I guess I got involved and how I can be useful to you. The other 3 minutes is we're going to talk about the problem statements and the judging criteria, and the last 3 we're going to start the team formation, or like basically team supplementation, if you will, if you already have a team. So just a brief background. I'm a bioengineer, also a computer scientist, roboticist. I have a really long description on my website. Apologize for that. But at least it gives you just an idea of maybe how it could be useful. My focus is, as an entrepreneur, is to use sensors and revisit, either older technologies or just even new technologies as well, and then they find the convergence with what's happening in digitization, for instance. I ran hackathons for FLIR systems, the thermal imaging camera, and I have it here with me just as an example of how neat, what an amazing time we live in right now. So this is actually piggybacking off of the same technology that CPUs and the GPUs are made off of, is the essentially etching process on silicon. So this little guy has 16,000 points, plus or minus 0.1 degrees Celsius. It basically can image in the IR spectra, the long wave IR spectra. And from any of you bio geeks can understand that in the non-visible spectra you can see a lot of interesting things. So one of the big things in deep learning is quality of data. And a lot of the things that we have is actually a lot of data. If we look at some of Dr. Giovanni's work and his colleagues doing family history and physical down to the molecular level as well, all that stuff is basically labeling that genome, right? So I just want to kind of really resound the fact that you do have a lot of data at the same time. My specific purpose is to make new data that's maybe specific to deep learning outcomes. So what we're trying to see is, with deep learning in general, is when you have this huge multivariate system like the human body, which is amazing, and also extremely complex and beautiful. The interesting thing is that if you perturbate that system like a proper engineer, you might be able to elucidate some interesting facts. So you can do some really neat stuff by doing very simple stuff to a very complex system. So I just wanted to touch on that and kind of get the creative juices flowing there. If you want to talk more about that, or if you need suggestions on interesting sensors, etc., we need to be nice to Ono. Make sure we don't non-invasive stuff, of course.
I'm ready to be, the subject of the experiment.
You're ready to be the guinea pig. That's awesome. Well, he gives up his data and he wants to be a guinea pig. That's just an amazing man right there. So again, just a brief thank you to everybody on the team. This has been an amazing experience to see this spin up in 2 months and what we've accomplished. So hopefully you guys get that energy that I experienced with this team. Real briefly on the logistics is that this is a mentor color. So if you see this color, just come ping us with any question whatsoever. No, the crazier the better, actually, because that's how innovation happens. So real briefly on the problem tracks first, and then I'll go into the judging criteria. So if you have one of these little flyers, we do want to kind of guide this whole effort towards these 4 basic points. So drug treatment pathways for existing medications. So this is something that hits close to Ono's project, or his mission, is to find other drugs out there that might be able to be candidates for Ono. And that's a huge thing in all rare cancers, because there's not that economic impetus like Recursive is talking about. But we need to understand that all cancers, all rare cancers, and all cancers in general, all rare cancers can also equal cancer in the deep learning realm, because what we're building is these frameworks to be able to solve and elucidate patterns. Again, so in a rare cancer type effort, you can actually find stuff for maybe a blockbuster drug as well. So I just encourage you to just really move forward in that regard. So ranking data set mutations. So again, most of the stuff is actually fairly obvious for your typical bioinformatician. If you have any questions, I would say just you go and ask one. If you guys have any questions, feel free. Use NLP to mine NF2 research and present novel treatment options. So then, create a novel drug intervention with DeepChem. So one of the other partner companies here are you making tools and tool sets for being able to find new solutions and new drugs. The judging criteria, if I can direct you to, I believe it's all on the website as well. So how well does the solution lead to positive outcomes for NF2 cancer patient treatment? Okay, well.
It is somewhere.
Yeah, no worries. Okay, so how novel is the approach to find? Yeah, exactly. It is. Yeah, let me actually address that. And how? Sorry. Who's on it? There we go. Cool. That's a lot easier. Just in case you don't have your laptop on there. So how well does the solution lead to positive outcomes for NF2 cancer patient treatment? So I'm not an NF2 expert in any way, and I've actually been out of the space for a while. I was part of kind of using SNPs and microarrays. We did some whole genome sequencing as well, but now it's interesting that some rare cancers eventually are going to show some interesting markers. So how novel is the approach to finding a solution? That's a fairly obvious question. Quality of depth of problem analysis, and then team dynamics, diversity, intersectionality. So this is the one that I really wanted to focus on number 4. Being a bioengineer, you're kind of like a generalist in many ways. I have some specialties. But what I encourage is that I saw a lot of hands go up for bioinformatics, and you have computer scientists as well. But what I wanted to throw out there now is team formation phase. Who out there has a very interesting job position that I think is pretty unique? Can you share with the crowd?
I scan people's brains for a living.
Fantastic. Okay, so he uses imaging modalities to find patterns as well, right? And
I do deep learning on it.
And do deep learning on it. Well, look, this is maybe somebody you want to have on your team.
Mentor.
Oh, okay, there we go. He's a mentor. You're right. I should pay attention to the color. So, anybody else out there that is looking for a team? Let's say. Maybe they want to. Okay, so hold up your hands up real high. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 17. Do we have enough time for 30 seconds apiece to give a little intro? Or?
Yeah, I would actually ask that all the team leads, we ask that you put your information on the spreadsheet that's in the Slack channel. So we do have an official team formation tab on the spreadsheet in the Slack channel under Hackathon Teams. So also, if you would like, if you're still looking for members to join your team, if the leader of your team can please come up, give a short description of specifically the problem set you're trying to solve, and then also what you're missing from your team, what skills you would still like to find. And then we could have you stand to the side, and then those that are going to be looking to join a team, we would have you come and meet with the person that you'd be looking to join. So can we have all the team leads please stand up right now? Team leads? So if you haven't chosen a team, that's fine, but team leads, and we can have you come up here one by one, please. Okay, yep, and then this is the spreadsheet that we're showing as well. So there's a second tab. So the first tab was a general, this is my name, this is what I know, this is what I'm looking for, contact me at. At the bottom, there's a second tab. This is the official Hackathon team registration. So it looks like we only have a couple teams officially registered so far. But again, we're asking that all of the leads, and whether you're registered or not, if you already know and have something in mind that you would like to do, if you can please come up, give a short description of the problem track you'd like to focus on, also the skill sets that you're looking for to join your team so that others can join you.
Hey, my name's Jeff. I'm here with my coworker Christian. We want to try using DeepChem to find a new drug intervention. We're looking for someone with systems biology experience or knowledge. That would be helpful.
Tell them about you.
My experience. I'm a senior data engineer in Silicon Valley Data Science. I have some experience with deep learning with Keras and TensorFlow, but mostly in computer vision applications.
And then stand to the side. And anyone that would like to join his team, please come over here and join his team. Can I have another team lead, please? Come on up. Yeah.
Hi, my name is Luke. I'm the founder of an AI startup, and we're going to do the NLP track. We're looking for people with Python, graph database, text parsing experience, any sort of NLP. So if that interests you, come find me on the left.
Okay, next.
Hi, I'm Guy. Fair warning, we're all undergraduates on my team, but we're thinking about doing ranking dataset mutations and also looking at unsupervised clustering methods, such as topological data analysis methods. If you're interested in mathematics, we're probably looking for people with a biomathematics background or also data wrangling, just in general. Thanks.
Okay, next team lead that's looking for people. Okay, how many people are not in a team and do not want to be a lead? Quite a few? Okay, great. Can you please join one of these teams or step up to be a lead to continue the hackathon as well, and then form with some other people. And then all the other teams that are currently formed, again, I want to remind you that we have a maximum of 6 people per team. So if you have more than 6 people, you have to kind of decide who may need to go to another team as well. And I need to remind you all to fill out this hackathon registration sheet. Okay, we're going to come around to all the teams tomorrow as well and just verify everyone's information. So it's very important that you're registered here.
Does anybody want to be a team lead? Anybody suddenly inspired to lead a team?
I'm not interested in being a team lead. However, whoever is doing drug treatment pathways for existing medications on your end, I don't know anything about it, but I want to.
That's the first guy. Yes.
I'll try to do the ranking dataset mutation.
Alright, looks like people are forming. Listen, if it's been 15, 20 minutes and you haven't found a team yet, come talk to one of us. We'll get you sorted out in a project or team or whatever. So don't let more than 15 or 20 minutes go by without talking to someone.